All right, let's go ahead and get started. Let's see, where are we today? Oh, I'm clicking. All right, so some just announcements. So if you can make it today for our poster session, our I guess makeup poster session, please come. I know we have a few stragglers, so there should be a little group today. But I know a few people are still kind of behind on things, so I am willing to do some sort of makeup thing next week as well. Sorry, trying to map my Wacom again. All right, week 10. And so the other thing that I would say, I feel like a lot of people are like falling behind. The general consensus I've heard is spring break or lack thereof really messed up everybody. But let's just finish strong. So if you need me, right, we're at the end. I was thinking, normally I have lab on Wednesdays as well from like 9 a.m. to noon. And we already finished up with Chem 104 though, so they're not going to be there. So that's just another good office hour time. I especially encourage you to just like make time with me to sit down and like finish assignments. You know, I feel like some people are really close, maybe just like missing a few post-lab things. Don't let small barriers prevent you from finishing. So just get her done. So let me know how I can help you. Like I said, willing to sit down whenever. I know you guys are busy and jobs and everything are probably starting to take off, but just try to finish strong. And then I know a few people are taking the exam early, so I'll probably send that over to the testing center. So make sure you talk to me about that if you want to take your exam early. Or if you need incomplete. So whichever way you're going, early or late, let me know. So we'll plan accordingly. Other questions, comments? We've made it to the end here. All right, so let's continue where we left off. And so we're about to start DNA and comparing it to a polymer. So, right, DNA is essentially our polymer. And so we'll look at how our double helix is built today. And so now we can look at essentially our monomers of DNA. And so our nucleotide is made up of three main components. So we have our sugar backbone, our phosphate group, and then some sort of base. And then we can appreciate the difference between DNA and RNA. And so here, so the two, right, deoxy, trying to pull up my highlighter here. And so the difference is just in the two position. So DNA has a hydrogen group, RNA has an hydroxy group there. So just kind of comparing the differences. And then you may have seen this in other classes or maybe high school biology or something. But I think most people are at least aware of our different bases. And so if you remember, we have adenine, guanine, and cytosine, and those occur in DNA and RNA. And you might remember something too of like who they pair with. And we'll look at why in just a second with their structure. But you might remember, okay, kind of the trick that maybe you're taught is the straight looking letters paired together. So A and T are complementary base pairs. And then the curvy ones go together. So G and C, so they're both kind of curved. So again, they form complementary base pairs. And then we also have, so uracil, so thymine is found in DNA. So just highlight that. So DNA, so T for DNA, and then U if we're in RNA. All right, so those are our 5 bases. And then we want to, again, just kind of appreciate the structure and be able to identify different pieces. So circling this, that's our phosphate group. Right, so identifying our phosphate group at every point there. We have our sugar is here. So just highlight your sugar. And then our base, right, is on the end there. So we could identify what our bases are. So it looks like this is A. Right, this is uracil. So etc. Right? Okay, so there's kind of one backbone. And so looking at this, right, this is kind of one side then to our double helix. So one strand. And so now we can see why these are complementary base pairs. And so looking at A and T, so they are able to form, right, 2 hydrogen bonds. So they're held together by hydrogen bonding interactions. So there's one pair. And then C and G are complementary since these form 3 hydrogen bonds. And so now that gives us basically our ladder, and so connects our 2 sides together. And so now let's look at our ends here. And so as it's drawn here, so this is what would be called our 5' end, and then 3', and then this would be the opposite. So this is 5' to 3', and so that's showing you the position that it's attached at the phosphorus and how it attaches. And again, it's kind of opposite, right? So it goes one direction one way, the opposite the other. And so let's kind of look at this. And so kind of, where are our notes? I was just debating. Let's see. All right, so let's kind of break this down, right? We have our notes. And so roughly we want to just understand, right? So we have, and this is just kind of our general overview, right? So we have DNA replication, where we're just, right, making more DNA. And so let's just kind of give an example of it and hit some of the highlights. And so DNA replication is called semi-conservative. And so let's look at our notes for kind of an example of how this would work. So we have, let's just say, our DNA strand. And so it's going to go 5' A, C, C, TCG AG dash 3 prime. And so then, right when we pair it, we just write the complementary strand. Except again, this is our 5 prime end. So sometimes when you look at like the answer, right, it'll be written in what almost looks like opposite, right, because it's starting at this 5 prime end. But then the main thing is you just want to be able to pair it, right? So with DNA, we have G with C, right? DNA is where we have T, so A with T, G with C, C with G, T with A, C with G, C with another G, and then T. So that's our 3 prime strain at the end here. And so like I said, right, if you saw the answer key of this, it might be written like so, but it's the same thing. So just looking at how you read it there. Okay, so then that's kind of one step of the process. And we won't go too much into it. I will say if you want to watch like good videos on this, Professor Dave, I don't know, are you guys familiar with Professor Dave Explains? Yeah. He does really good videos on this with like the, you know, the little cartoons and everything. And so he does like a much better job of kind of explaining how exactly it's synthesized. I don't think I have a good visual because it's kind of confusing because you have like a leading strand that's synthesized first and then a lagging strand that goes like piecewise. But I think it'd be, I could probably post a link to this. So Professor Dave, like I said, does a much better job with the little cartoon graphics to help. All right, so that's at least kind of the brief explanation of DNA. And so then we have, right, we want to get to, well, how do we make proteins, right? So that's kind of like where this is eventually going, is we have our DNA. And, well, if we want to make a protein, well, okay, well, now we copied our DNA. But we need to have our DNA go to RNA, right, so that eventually this RNA can go out and code for the protein. Right, so send RNA for protein. So this is where we're trying to go back to our amino acid relationship, basically. And so again, pretty similar, except now the RNA, right, uses the U. So we have uracil instead of the thymine. So then you can code. And so that process is transcription of DNA to RNA. And then going back and relating it back to proteins is the last step is, right, translating. So it's called translation. So translating back, right, that RNA code and now making it a protein. And so then we can go to our notes. So we'll skip our sugars for a second. And so that's what these charts are for. And so you start, I think this one is kind of the easiest one to read, right? So you start in the middle, so there's your 5' N, and then you just go with your sequence, right? So depending on what you're starting with. And then you can see each 3 codes for a specific amino acid, and then some of them also code for stop and start codons, so that they know where to start synthesizing the protein. Sorry, looking. So I think this is from your homework. And so let's just start with, let's look at number 6. The complementary mRNA strand. Yeah, sorry. So then, I wasn't slick either. So what is the complementary mRNA strand of our DNA? And so 5ACCTCGAG 3 prime. All right, so what's our complementary strand? So then looking at this one, right, you write it so 5 prime, and then I'm just matching initially, right? So C, U, C, G, A, G, G, U. And then, right, again, this is where the answer key probably rewrites it as 5 prime, right? C, U, C, G, A, G, G, U there. So same thing, just kind of rewritten, right, in this 5 prime order. And so then we can, again, take this mRNA now and translate it. So we could, let me write out, so we could have AUG, GCA. And so starting at the middle, see if you can identify what amino acids those correspond to. I can scroll up and down. They should have the same information. They just have different keys. think this one might be a better chart, actually. I'm going to repost it. This one's kind of easier to read. All right, so what do we find? So what did AUG code for? So start or methionine. And then GCA, alanine, UUU. Yeah, phenylalanine. And then last one. So stop. So good. And as you probably noticed, right, there's several different combinations to code, right, for one amino acid, but this combination only codes for one amino acid, right? It won't code for other things. So. All right, let's shift gears then. Let me move. I don't know how my glucose galactose got shifted. All right. So our last bit of this kind of hodgepodge chapter is our starches. And so starches are also considered polymers, right? They're just polymers of sugar. And once again, oops, sorry, I put this on the wrong place. And so they're formed just like we've seen with our other polymers from our condensation reaction. So losing water. And so that's what's shown here. So we have our glucose and our fructose. And so they can come together again with a condensation reaction. Already highlighted or in green is again what's going to leave and end up making water. So you can see that bond form. And then sugars have different forms and different ways that they can bond to one another. So if you have an alpha form, we have these two hydroxy groups are on the same side of the ring. So notice how they're both kind of pointed down versus the beta form where they're both on opposite sides. And you can kind of see it going opposite. And so other things, there are other forms to look at these sugars. And so you often see them used interchangeably. And so you have two forms of your sugar, and so they're interchangeable through this process called tautomerization. And so they're just kind of changing forms. And so you have the, what would be called the pyranose form. And this is our ring form of our sugar. And then it makes our open chain and then reforms this smaller ring of our furanose. And so with that, you often see what's called our Fisher projection. And so let's look at how to convert a Fisher projection to Hayworth, is what it's called. And maybe I'll get a picture. Let me see if I can find D-glucose before I have to draw out this ring. Alright, so luckily if we look up D-glucose, it's usually shown in this kind of ring form here. So let's copy this. Alright, so we have our D-glucose here. And so now I'll draw out what's called our Fisher projection. And so your Fisher projection looks kind of weird. It's like this kind of backbone looking thing. So we'll draw it out and then we'll translate it. So we have our CHO and OH, hydrogen. So this is first carbon, second carbon, third carbon, fourth carbon, fifth, and then our sixth down here. And then translating that. So now we have 1, 2, 3, 4, 5, then 6 up here. Right, so there are two different forms. And then so kind of there's a couple of tricks that people kind of use to help you translate this. It's okay, just kind of staring at these. And so looking at them, so we have 2, 3, and 4. So carbon, right, 2, 3, and 4. They're right side down. And kind of what also people say is like you can think about the stereochemistry as them like hugging one another is how they often say. And so kind of like trying to translate it is imagine, so now I'm just going to like redraw it. So we have, again, so they're all like hugging and coming out. So trying to draw our stereochemistry. Since that's what we care about with our sugars is if we're up or down with these hydroxy groups. So just kind of drawing, redrawing. So everything's right kind of coming out, hugging. And then you can kind of rotate and tumble it. And again, that kind of gives you a better picture of what it looks like. So now this is coming out, and now this is down, right? So this is 1, 2. So now you can see, okay, 2, right, is down, and keep going. So C, O, H, H, right? So 3 is up, right? So you can see that 3 is up. So 4, hydroxy down. CH2OH, so 2, 3, 4, 5, and then so going back, right, 4, 5, so they're tumbled down. So you can kind of translate that. All right, so let's look at then, I think this is from your homework, right? Yeah, I was just trying to pull up the copy. So I guess the question that it asks something if they are identical or not, correct? Yeah, so they're different. So at carbon 4, right, they got one's up, the other's down. So they're different. And uh, then it asks if they are, what does it ask actually? I still haven't pulled up the homework. Okay, so then are they isomers or optical isomers? So what do we think? So yes, they're optical isomers. So what do you guys get for that? So is it a mirror image though? Because it's not perfectly a mirror image, right? It's only flipped though at one stereo center. So these are diastereomers. But yeah, not a true. So an enantiomer, right, would be a mirror image, but it's not fully, right, complete mirror image, just, again, flipped at one stereo center. But still optical isomers. And then again, we want to write our glucoses and distinguish between the alpha and the beta glucose like we were looking at. And so kind of going back to this Hayworth projection, this is, again, often used to kind of show differences. So let's draw out, and this might have been your homework too as well, to draw out the difference between alpha and beta glucose. And so it's much simpler if you draw it, like I said, in this Hayworth projection. And so let's start off with alpha D glucose. And so we'll make our ring. So our 6-membered ring. So oxygen, 5, 6 there. And CH2OH. And then this one, right, is going down. So that's where it's important. And where this projection, I think, kind of helps, hopefully. Hopefully it helps people with the up-down. And so alpha, I think you just said it, but was the same side. And so there we're looking at our 1 and 4 carbons. So 1 and 4. So again, those groups off of them are on the same side. And then the advantage of this form of drawing it in this projection is that it's easy enough to do the beta form. So now beta D-glucose is just now these are going to be opposite. And so now we can just copy our backbone here. Oops, I did it wrong. Sorry, I wasn't paying attention. So I meant to flip this one. So this one goes up, and then this 4-1 can stay down. So everything else is the same, right? All these other hydrogens are the same. We just flipped the 1. Yeah. I'm not like 100%. I'm like not the greatest sugar chemist because I feel like if we flip the other one, I would have to just double check that we didn't make the L isomer or something crazy. But yeah, I'm not the greatest sugar chemist. I'd have to like stare at some pictures. Otherwise, it shouldn't matter for our purposes, right? As long as they're the opposite, then I'll understand that you're doing beta. But all right, we have some time, so let's. Were there any other questions on the homework, by the way? I never pulled up the homework, sorry. It was slow instead of slick on my computer. But did we encounter any other kind of issues with the homework or anything before? I wanted to do the demo for our last bits. Like which one? Oh. I don't remember if I tested you on that, but I mean, it seems like a fair question, right? Of finding like a chiral center. Just maybe not as complex. We didn't do this one. We could do it right now. Or just check it out at least. So number 4, where it says draw the structure. So I don't know if you want to do that one of the tetrapeptide. So met. So you're going to have something similar like this on the exam, except I probably won't make you draw it out, but what you'll do is kind of, I'll say like, this is the amino acid it's supposed to be, or like the chain, and then here's it drawn, is it drawn correctly type of thing. So I don't know if you want to do this or just kind of know to pay attention to it on your homework. If we don't want to like do it, I would just say like go back to the backbone that we talked about. If I can find it now. Sorry, I was trying to find, where's our cheat sheet? Okay, this is our better cheat sheet, right? Like this is the easy way to draw any generic peptide chain, so amino acid chain, right? So just watch for that. Watch all your connectivities. Trying to think of if there's anything else. I probably won't go sugar heavy. Let me look at your exam. Just trying to remember what else is on there. But yeah, just trying to stick to big picture ideas at the end. And just looking at like the main kind of DNA complementary strains, and we don't need to get too detailed into the process of it. So yeah, I think I stuck away from the sugar stuff. I do have a hybridization question. So I don't have like an identifying stereo centers right now, but I do have it as like, what's the hybridization on a molecule? But yeah, yeah. I'm just kind of looking at the final exam and what's on there. But yeah, there's a tetrapeptide, like I'd mentioned, that you have to see is it drawn correctly or not. So we'll get at least look over this. Like I said, we could do it, but I think you're better off just like staring at it on your own for a little bit and writing and practicing. And then there's, a bit of the polymerization. I'm just looking at your exam. And then, yeah, a little bit of like writing the balanced nuclear equation. So hopefully that's not too bad. We'll do some half-life practice on Tuesday probably. There's some ligand stuff of high spin, low spin. All right, that's probably a good coverage. So let's skip over to the demo. Unless, like I said, unless there are other questions. Okay, trying to get my camera working. There we go. Alright, so I promised y'all I would do the nylon demo. So let's look at what I brought today for chemicals. So we're doing, like I said, I promised the nylon 6,6. And I have slightly different compounds than I think we went over. So I brought hexamine diamine, like I promised. 1, 2, 3, 4, 5, 6. So 1,6-hexane diamine. And instead of an alcohol or carboxylic acid, I got something slightly different but similar of adipole chloride, which I will draw out for you. And so instead of having an OH here, it has a chlorine. And so what's going to be leaving then, do you think, instead of water? So chlorine, hint, hint. Yeah, but try again. So you have an OH here originally that made water. So no, because remember it breaks this thing. 1, 2, 3, 4, 5. Yeah. So then we make a little bit of HCl when we do this. Essentially condensation reaction, right? So similar mechanism of our nitrogen is going to be attacking, right, our carbon, our electrophilic carbon again, and it'll be same, right, sort of mechanism, except exactly we're going to have HCl leave for our condensation reaction. All right, so let's try that. And so I'll have to, after we make the nylon, I'll run off and rinse it, since obviously I'll have a bunch of HCl on it. And then once it's rinsed, we can pass it around. And so once I'm done with the demo, I'll hand out the quiz, but make sure you sign in for the quiz. So I decided, I wasn't sure how people were feeling, if y'all were feeling confident on these. And I looked at the quiz and I was a little worried. So I figured maybe we should just use it as like exam prep. So you'll get full credit for coming, and then you can practice the quiz. And then I don't know if you want to go over it probably on Tuesday when we do the review. And at least it'll give you an idea of what to study so that you feel comfortable with the types of questions. Oh, I forgot my sodium hydroxide. I don't know how I forgot that. I'll grab that in a second. Too much ADD. All right, so let's at least start it. Like I said, I'm going to have to grab sodium hydroxide unless any fairies hear me, magical stockroom fairies. All right, so we need about 8 mils of our hexamethylene diamine, and it's in a 5% aqueous solution. So it's already in a aqueous solution, so I'm just going to guesstimate about 8 mils and make sure to not contaminate my flask. So they have to be dry. Okay, that's probably good enough. All right, so I'll run and get some sodium hydroxide in a second. Before I do that, so we have our adipole chloride. And so I'll take about, trying to get just, oh, it's slightly degrading. You can tell it's just a little yellowy, a little nasty. All right, so half a mil of that, and then I'm just going to add to it cyclohexane is the solvent that we're just diluting it in. Oh, I knew I should have brought a smaller bottle. Okay, let me grab 2 things. I didn't want to run around with this with like an open beaker, but they didn't have a smaller bottle for me. So let me grab a beaker and the sodium hydroxide. I'll be right back. All right, so I'll add 8. So I added sodium hydroxide to the hexamine diamine. Then I just need to pour out my cyclohexane a little. All right, so dilute it. A few mils, about 8 mils. All right, so let's try that. And so now all we're going to do is pipette in our cyclohexane solution. And then you should see it start to form kind of this layer. So I'm just... And then you can actually see it already. So you can always get closer too. But you see how the flask is already a little cloudy? And so right at the point where the two liquids are meeting, we're forming our polymer. I forgot one more thing. Oops. I'm just adding it all in here. So now you can grab it out. And so you can see it forming. And so the one thing that I forgot was tweezers. And so normally you can kind of pull this out with tweezers and just kind of keep going. So it's formed another little layer. So there's our nylon. All right, I'm going to stop there for now. So I'll probably run to the stock room and just wash it up a little, and then we can pass around the nylon if you're interested, just to see what it looks like. And so again, right, most of your fabrics now are synthetic, right? You have nylon. I think now there's what, rayon. I'm missing, what's that new one that's come out? Now it's eluding me, but there's plenty of synthetic fabrics. So yeah, try the quiz. I would try the quiz on your own as to kind of gauge right where you're at personally and how much studying you need to do. And then as usual, right, just get everything in by the end of the quarter. So ideally by next Friday, but as you all probably know, I'll accept things up to like next Sunday, right? It's not this Sunday, but a week from now. What is that, like June? So we won't be collecting that. So yeah, like I said, sign in. That's your grade. And then we'll go over the answers probably on Tuesday. All right, any other questions? I'll leave the stream going for a second in case there are questions. I'll go wash this and I'll be back.